Now we are going to study the interferons and interleukins. What is the interferon? Maybe these are some of the antiviral agents. The people who have been suffering from the hepatitis B or hepatitis C, they have heard this name of the interferons very clearly. We use them as the antiviral therapy. We use them for the supporting against the hepatitis B or hepatitis C or some viral diseases. We use the interferons. So let's see them in detail. These are the natural antiviral agents. These are produced inside the body when any pathogen or the virus attacks the host cell. These are the actually host immune systems. Interleukins and interferons are the basic parts of the host immune systems. Substances that have the antiviral properties in the adjacent and the non-infected cells are called interferons. Here you can see their structure of the interferons. Types of the interferons. At this moment we have about two types of the interferons. First of all are the interferon alpha, beta, and other one is the interferon gamma. All of them have the antiviral activity. The maximum antiviral activity are combined with the interferon alpha. However, the interferon beta can also give us the antiviral activities, but to a less amount, less extent, and they are the intermediate antiviral activity. Then the interferon gamma are the more lymphokine than the antiviral alpha and the antiviral beta. Regulation of the interferon expressions. The interferon is not expressed in the normal resting cells. They will be expressed until the immune response is awakened, until the attack of any pathogen, and it depends upon the specificity of the interferon, that what kind of the pathogen is attacked, what should be the release from the immune system, whether this should be the interferons or the interleukins, or what kind of the response should be shown by the specific by the immune responses. Labile precursor binds to the protein elements and the blocks transcription and through the interferons. Its production of the labile repressors dropped in the viral infection and allows the interferon synthesis to occur. Here you can see the interferons, the mechanism through which the interferons are released inside the body with the action of the virus. The virus attack, virus nucleic acid will be entered inside the body. Here is the interferon genes are turned on after the virus will be in replicating inside the body. Here is the DNA, the viral DNA and mRNA, and virus is going to replicate inside the nucleus, and the interferon molecules will be awoken and they will be released from the body. Here are the interferons. The host cell infected by the virus makes the interferon, is killed by the virus. Here is the host cell 2 where we are converting the interferons. Entered by the interferon from cell 1 induces the changes that to protect it. Interferon stimulates cell to turn on the genes for the antiviral proteins. So therefore the antiviral proteins block the viral reproduction. The basic function of the interferons is to produce the antiviral proteins, and these antiviral proteins will act to block the viral infection. Just like from this diagram, it's clear that here are the interferons are produced at the end of the infection. The viral mRNA is already replicating. However, if we give the interferons at the time of the infection and then the proteins will be produced, those will be the antiviral proteins and which are capable of block the viral reproduction. The clinical uses of the interferons. Interferon A is used for the treatment of the viral infections. Most basically the viruses which are used are the Condylomata acuminata and the chronic hepatitis B and C viruses. We can use the interferon A. These are also used for the prophylactic and therapeutic agent in the immunocompromising diseases, just like HSV, VZV, or the other, just like similar type of the diseases, herpes virus 1 or the herpes virus 2, we use the interferon A. Then they can also be used as the prophylaxis for the CMV in the renal transplant. They are also used, interferon A is also used for the treatment of the AIDS associated with Kaposi sarcoma and hairy cell leukemia. Then there is the interferon G, immunostimulant in the oncology and immunodeficiency syndromes.